Hello everyone. I am one of the biology faculty member at Education World Gurdaspur, one of the famous and renowned institutes in the city Gurdaspur of Punjab. And in this presentation I am discussing on the theme cell. This is my second lecture with concern to the 9th ICSE biology, and I have started discussing the first chapter, basics of biology, the cell. And if I will recapitulate my previous lecture with regard to the cell, then we have discussed already. The cell is defined as the structural as well as a functional unit of life. This is a fundamental unit of life, that is, the bodies of all the living species is comprised of cells, and even the functionality of each and every species is determined by its constituent cells. In other words, I would want to say the activities shown by an organism is actually due to the sum total of the activities shown by its constituent cells. And you may see here the picture. The various species are shown here. Some species belongs to the animals and some they belongs to the plants and other ones also. And you may see what about the unity in diversity at the cellular level. We can say that the bodies of all the living creatures, of all the living species is comprised of cell. This is the unity in diversity. In other words, cell is the link between the various species. With regard to the discovery of the cell, Robert Hooke first of all discovered the cell, and he discovered the cell from the cork cells with the help of self-designed microscope. He observed the cork cells and he was credited with the discovery of the cell. But for your kind information, cork cells are the dead cells. The live cell was first of all visualized by Anton von Leeuwenhoek and by his self-designed microscope. Leeuwenhoek done a number of discoveries in the field of cell biology. He was a Dutch lens maker. He was not a scientist or biologist from the origin point, but he has curiosity with regard to the certain living materials, and his curiosity enabled him to discover the various types of living species, the various types of living cells. For example, Anton von Leeuwenhoek collected the bacteria from the rainwater, from the tartar of the teeth, from the curd sample, and he was the first man to observe the bacteria. He was the man who discovered the bacteria, and he is also known as the father of bacteria. Then, Leeuwenhoek also discovered the RBC. He made the blood smear and observed the red cells in it and named them as RBC. He also discovered the first protozoan, Giardia intestinalis, which is commonly called as old grandman of intestine. The first pathogenic protozoan discovered by Anton von Leeuwenhoek. And in addition to that, he also observed the spermatozoon or sperm. And now in the current era, we are having the sophisticated instrumentation which helps us to have the deep insight into the cell composition, cell structure and cell function. The two commonly employed instruments in this regard are, one is the compound microscope and second is the electron microscope. Compound microscope is also known as a light microscope. It is also referred with the name of student microscope, and this is the most commonly employed microscope in the laboratories as well as in the school or colleges and even in the universities. But in the university level, you may also see a bigger microscope, which is the electron microscope. And the major difference I have already discussed in the previous lecture. The light microscope utilizes the visible light as a source of illumination, whereas the electron microscope used electron beam in order to visualize the object. Then in the compound microscope, we use optical lenses like eyepiece or ocular, then the objective and condenser lens, whereas in case of electron microscope, we are using the electromagnetic lenses. Compound microscope has low magnification power, whereas the magnification power of electron microscope is in lakhs. So further, advancements in the field of cell biology was made by Schleiden and Schwann. Schleiden was a German botanist and Schwann was a German zoologist, and both of these two reported that the plants and animals are comprised of small compartments known as cells. And they proposed a theory which is referred to the name of cell theory. And the major conclusion of the cell theory was the bodies of all organisms, that is the plants and animals, are composed of cells and cell products. Then the Rodolphe Virchow in the year 1855 discovered a new property of the cell, that is omnis celluli cellula, that is the new cells they are formed by the division of pre-existing cells. And he also modified the cell theory that was given by the Schleiden and Schwann, and the new theory was referred to the name of modern cell theory. So normally if we see the cell, a cell is primarily made up of two different kinds of substances: cytoplasm, which is the fluid material present inside the cell, and the organelles. So now, if we observe the cell with respect to their size, so the cells vary in their size from smallest mycoplasma, which is having the size range 0.3 micrometer, to the cells which are in millimeter, like ostrich egg. The largest cell is the egg of ostrich, the longest cell is the nerve cell, and the smallest human cell is the RBC. It is having 7 micrometer size with respect to its diameter. On the basis of their shape, the cells are of various types, like they may be disc-like, they may be polygonal, they may be cylindrical, they may be irregular, they may be thread-like, or they may be elongated. These are the various different shapes of the cells which we can have in the different life forms. And primarily, the cells are of two types: prokaryotic cell and eukaryotic cells. Prokaryotic cells refers to the bacterial cells. Prokaryotic cells refers to bacterial cells, whereas the eukaryotic cells refers to plant cell, animal cell, fungal cell, protist cell, etc. So if we discuss today about a little bit about prokaryotes or prokaryotic cells, so prokaryotic cells are those cells which are characterized by the presence of false nucleus. That is, they don't possess the true nucleus, but they will possess the false nucleus. False nucleus in the sense that the nucleus is without any covering membrane. In addition to that, the prokaryotes, they don't have any membrane-bound organelles in them, and they include bacteria, blue-green algae, mycoplasma, PPLO, that is the pleuropneumonia-like organisms, and they are generally smaller in size, they multiply more rapidly than the eukaryotic cell, and they also vary in their shape as well as the sizes. So you may see the various kinds of prokaryotes. like bacteria are there, cyanobacteria, the blue-green algae is there, mycoplasma, which is also known as a PPL or pleuropneumonia-like organism. All of these, they are the prokaryotes. The prokaryotes, they have the cell wall which is made up of a special kind of material which is called as peptidoglycan, which is also known as a murin or mucopeptide content. And they are usually immortal because the natural death is not there. They divide again and again, and they have a huge capability for asexual reproduction, primarily binary fission. If we observe the shapes of the prokaryotic cell, they occur in the variable shapes. They may be spherical in outline, as we report in case of coccus bacteria. They may be rod-shaped in outline. This shape is referred to the name of bacillus, and the bacteria is known as a bacillus bacteria. Likewise, they may be having a spirally coiled shape. This is called as a spirillum bacteria. Okay, they may be coma-shaped in outline. They may be referred as a vibrio bacteria, such as we can exemplify with the vibrio cholera bacteria. The prokaryotic cell has only single envelope type of organization. That is, they have only a single envelope in the form of we can say that cell wall or cell membrane around the cell, but the membrane is absent around the nucleus, as well as the membrane bound organelles, they are absent. So furthermore, they have a few specific organelles such as the mesosome, chromatophores, nucleoid, flagella, villi and fimbriae, ribosomes, and certain inclusion bodies. So in the next lecture, I will discuss with you with regard to the various kinds of the cell organelles, as well which are reported in the prokaryotic cell, along with the details of prokaryotic cell. Thank you so much. If you like this video, kindly subscribe our Education World EW YouTube channel and share this video, and you may press the bell icon button in order to get the notification for the lectures which have been uploaded from time to time. Thank you so much.